Hi, my name is Sévoz-Couche, and I am a research scientist from INSERM institution. And I will talk about the vulnerability to depression and the occurrence of cardiac events with a focus on the vagus nerve. So during stress, there is activation of the hypothalamus and the hypophysis and adrenal gland. So this is the HPA axis, leading to the release of cortisol, influencing the brain, but also influencing the heart and at the origin of cardiovascular disease such as coronary heart disease. But there is also the occurrence of ventricular fibrillation. And we have asked whether this could be due to the inhibition of the cardiovagal efferents innervating the heart. So to answer this question, we have performed experiments with an animal model of stress called social defeat. And this model is a protocol of stress divided into two phases, one phase of anticipation followed by a phase of submission. And after this protocol, we have waited 4 weeks, and then during 2 weeks, we have performed chronic mild stress. And at day 45, we were able to identify two groups of animals. Some of them were non-vulnerable to depression, and some others were vulnerable to depression compared to non-defeated rats. So the question was to know whether the vagal function was reduced in vulnerable animals compared to non-vulnerable animals. So we have implanted captors, telemetric captors in rats, and we have analyzed heart rate variability and especially the high frequency variations, because this is under the influence of the vagus nerve and reflects cardiac vagal function. And we have shown that after a few days, 5-6 days after social defeat, both groups of defeated animals, non-vulnerable and vulnerable to depression, had a reduced vagal function compared to non-defeated animals. And then around day 20, then some rats recovered their vagal function, and these animals were those non-vulnerable to depression, while the animals vulnerable to depression kept reduced vagal function until day 30. And when we looked at the occurrence of ventricular premature beats in these animals, we had exactly the same timeline in mirror to the vagal function, because at day 10, so 5-6 days after social defeat, we had the occurrence of ventricular premature beats in both groups of vulnerable and non-vulnerable animals. And then again, around day 20, vulnerable animals kept a high number of ventricular premature beats, while the non-vulnerable animals tended to recover until day 30. So, in conclusion, social defeat induces vulnerability to depression, and vulnerability to depression is associated with a persistent decrease in vagal function, synchronized with an increase in ventricular premature beats. Thank you.